Hello, microbiologists. So we have just finished our discussion of prokaryotic cell anatomy, and now we're going to turn our attention to eukaryotic cells. These typically tend to be cells that are larger, and because they're larger, they are more complex. So just like if you think about like a little company that's run out of somebody's garage, probably doesn't have the complexity compared to like a Fortune 500 company that's going to have lots of different departments and different organizations within that company. This is kind of the eukaryotic cell is the bigger company, so it has more complexity to it. So eukaryotic cells can have flagella. So here's a flagella in this cell. They can also have cilia. Even human cells, there are human cells like sperm cells that have flagella. You have cells in your airway that have cilia on them. In eukaryotes, flagella and cilia tend to be projections of the cell membrane and contain cytoplasm. Although they're still used for locomotion, they may have one or a few flagella. Cilia are going to be more numerous and shorter. And opposed to prokaryotic flagella, which rotate in a circular motion, eukaryotic flagella are going to move more in a wave. Eukaryotic cells sometimes will have a cell wall. They are typically going to be simpler than they are in prokaryotes. So where prokaryotes had more complicated structures of different types of sugars and proteins, in a eukaryote, it's typically going to be a carbohydrate like chitin or cellulose. Plants, algae, and fungi all have cell walls in them. In animal cells, we don't have a cell wall, but we do have a glycocalyx. A glycocalyx is a more slimy, loosely organized layer of carbohydrates that are outside of a plasma membrane. It will be bonded to the proteins and lipids within the cell membrane to stay attached to the cell. The plasma membrane in eukaryotes is going to be similar to the structure and the functions of prokaryotes. We may have some different proteins. We also have different carbohydrates that serve as attachment sites. One thing that's important in a eukaryotic cell is being able to have cells attached together to organize into tissues and organs. Eukaryotic cells tend to have sterols within their membrane, which are a kind of fat. We discussed these in chapter 2. They help resist lysis of the cell. We don't have group translocation occurring, although we do have simple diffusion, facilitated diffusion, active transport. And another type of active transport we have in eukaryotic cells is called endocytosis. This is a way to bring things into the cell, such as phagocytosis, where a cell can bring in food particles, or pinocytosis, where a food can bring in liquid from its environment. The eukaryotic cell still has cytoplasm. This is the stuff that's inside of the plasma membrane. However, it's technically not what's in the nucleus. The cytoplasm is technically outside of the nucleus, inside the plasma membrane. Cytosol and cytoplasm are two words that you can use synonymously. I'm not going to be that picky. It's the wet stuff inside of your cell. Inside of the cell is also a cytoskeleton. So just like your skeleton gives structure to your body, kind of controls the shape and movement of your cell, the cytoskeleton is going to give structure and control the shape and movement of a cell. These are made of filaments and microtubules. They not only allow your cell to move, they also transport things inside the cell. So they act as kind of a little bit of a highway system inside the cell to allow things to get where they need to. So you'll see something called cytoplasmic streaming, where the cytoplasm can move throughout the cell. This allows nutrients to be dispersed throughout the entire cell. Instead of staying concentrated on one end of the cell, it's distributed through the whole cell, so the whole cell maintains the energy and materials that it needs. Eukaryotic cells also have ribosomes, and again, this is not math, because this doesn't math. This is telling you where, if you centrifuge the cell, where the different subunits would end up. It's a measurement of both size and also shape. So you have a 40S subunit and a 60S subunit. These are the two subunits of a eukaryotic ribosome, which together make an 80S ribosome. These are different numbers than what you saw in a prokaryotic cell. So you can tell the difference between if it's eukaryotic or prokaryotic, because a eukaryotic cell has an 80S ribosome and a prokaryotic cell has a 70S ribosome. Now, in a prokaryotic cell, all of the ribosomes are floating throughout the cytosol inside the cell. In a prokaryotic cell, some of the ribosomes could still be floating throughout the cytoplasm unattached to any structure. These are called free ribosomes. But some ribosomes are going to start attaching to some of these membrane organelles. Fixed ribosomes can attach to the endoplasmic reticulum or to the envelope of the nucleus. So you can see ribosomes attached to both of these materials inside a cell. And where the ribosome is located will determine what kind of protein it's making. So if a ribosome is making proteins for use to just float inside the cell, it will be a free ribosome. And if a ribosome is making proteins that need to be stuck in the plasma membrane, or that need to be secreted from the cell, or need to go to a lysosome, those proteins are going to be made on fixed ribosomes. Fixed ribosomes and free ribosomes are completely identical except for where they're located. So just like you're the same person whether you're at school or at work or at home, the only difference is where you are. Same thing for fixed and free ribosomes. They're in different places, and in different places they do different things, but they're still the exact same thing. So at this point, tell me how are flagella and cilia similar or different in eukaryotes and prokaryotes? Cell walls, how are they different in eukaryotes and prokaryotes? Plasma membranes and cytoplasm, what's a way that they are similar or different in eukaryotes or prokaryotes? Then we'll finish up by talking about our organelles. Organelles are like the departments within a Fortune 500 business. These are specific parts of the cell. They have specific shapes and specific functions, and they are separated from the rest of the cytoplasm by a membrane around them. Membrane-bound organelles are only found in eukaryotic cells, and they include the nucleus, the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, mitochondria, and chloroplasts. I would like you to be able to briefly explain, maybe, one sentence, tell me what each of these is doing. Or if I give you a one sentence explanation, what organelle does this, you should be able to tell me that. Okay, so the nucleus is the biggest, most obvious structure within a cell. If you're looking at it in a microscope, almost always you'll be able to see what the microscope is. It is where all of your DNA is stored. So this is really the boss's office. It has the rules for determining the cell's shape, determining the cell structure, determining how protein synthesis is going to be going on. It has DNA, which is the hereditary material, the information you inherited from your parents. This is where your chromosomes are. Specific parts of the nucleus, terms that you should also be able to describe to me. First is the nucleolus. The nucleolus is where ribosomal RNA is being made. So if your cell needs a lot of ribosomes, it needs to make a lot of proteins, you're going to have a very busy, large nucleolus. And if your cell is not needing to make ribosomes, you won't have such an active nucleolus. Chromatin is kind of all dispersed through this whole thing. Chromatin is just loose, thread-like DNA. When your cell is at rest, not dividing, its DNA is all spread out and available to be used. The nuclear envelope is this material that surrounds the nucleus. It isolates the nucleus and separates it from what's going on outside in the rest of the cell. And it's a double membrane. So it has a bilayer here and a bilayer here. So you have 2 layers of phospholipids and 2 layers of phospholipids. So altogether, there's 4 layers of phospholipids. And there are also little pores, these little holes that allow things to go in and out of the nucleus. So if your cell receives a signal, it can send that signal into the nucleus. And if the nucleus makes some sort of material or product, it can send it out into the cytosol. The endoplasmic reticulum is a network of membranes connected to the nuclear envelope. So all these it looks like just a bunch of paper plates stacked on top of each other. And it does things like make proteins, carbohydrates, and lipids. It stores those molecules. It transports materials within the ER. And it also detoxifies drugs or toxins. So someone who is a little bit crazier and spent the night engaging in illicit substances would have a very busy ER the next day as they were detoxifying all of those materials that I'm sure you all haven't even heard of. You're such good kids. There's two types of endoplasmic reticulum, smooth ER and rough ER. The rough ER is called rough because it's got all these little bumpies on it. It's got all these ribosomes attached to it. And the smooth ER is nice and smooth. It doesn't have all those bumps all over it. Then to get from the ER to the Golgi, your endoplasmic reticulum is going to make these little vesicles. So vesicles are the little Ubers that will take whatever you made in your ER, take it to the Golgi. In the Golgi, you have another set of, phospholipid paper plates. They're also going to be near the nucleus, and they will take things from the ER. So these secretory vesicles are coming from the ER. They're going to come and merge with the Golgi and just kind of make its way from the cis face on down to the trans face. While it's going through these layers, it's going to repackage the secretions, package them into specific vesicles, and then the trans face will release transport vesicles to be able to go off. And these transport vesicles will go three places. One is it could be released out of the cell. So here you see this one kind of being secreted, and you see it just being pushed out of the cell. Maybe it's a hormone or some signal that needs to be sent to other cells. One is it could be part of the plasma membrane. So you see this vesicle coming, and it's just merging with the membrane because this is just providing new phospholipids for the membrane. And one is it could remain in the cytosol and mature into a lysosome. So this one here, you can see this vesicle is gathering the enzymes to become a lysosome and maturing into a lysosome. So we saw a membrane flowing from the nucleus to the endoplasmic reticulum, to the Golgi apparatus, to vesicles, to the plasma membrane. That's how our membrane is going to flow from the nucleus to the endoplasmic reticulum, to the Golgi, to the plasma membrane. So this continuous flow, this continuous movement of membranes using vesicles can allow your cell to replace the plasma membrane, can allow you to put new molecules in your plasma membrane, so your cell can adapt and change. It's constantly going in this direction only. Nucleus, ER, Golgi, plasma membrane. Be able to tell me this direction of membrane flow. Tell me what it is and what it's used for. Mitochondria are the site of energy production. It's where you make ATP. We'll come back and talk about mitochondria a lot. For right now, you know they are the energy powerhouse of the cell. It's where you're making ATP. Peroxisomes are going to help break down fatty acids, and they make hydrogen peroxide. They help oxidize things that you don't need around your cell, so they're helping break down stuff that you don't need. Vacuoles are spaces in the cytoplasm, especially in plant cells or fungal cells. They can hold water. They can hold waste or poisons. As they take up water, they can get bigger, so they allow your cell to get kind of more turgid and firm. So they provide some rigidity to leaves and stems. If their vacuoles are full of water, that plant cell is going to be a little more rigid, and that flower will stand up tall. As it starts losing water, if it hasn't been watered lately, that vacuole will shrink down and the flower will kind of just droop. Chloroplasts are found in algae cells and in plant cells. They are not in animal or fungal cells, though. They have the pigment chlorophyll, and they're used in photosynthesis. They look like stacks of macarons here, little cookies or something. And these little sacs are called thylakoids, and the whole stack is called a granum. Okay, craziest thing we talk about this semester is how our eukaryotes evolved the organelles that we have. So first life on Earth were simple organisms, eukaryotes, about 4 billion years ago. First eukaryotes evolved about 2.5 billion years ago. So for a long time, life was dominated by only prokaryotes. As we started having eukaryotic cells, a lot of eukaryotic cells got energy by eating other smaller bacterial cells. But at one point, we had a eukaryotic cell that ate another cell, but it didn't actually eat it. It kept that cell inside and developed a little symbiotic relationship with it. Eventually, that little cell became a mitochondria. And as crazy as it is that it happened once, it happened a second time. A cell with a mitochondria ingested another cell, but instead of eating it, developed a symbiotic relationship, and this little bacteria became a chloroplast. If you were to look at a cell, mitochondria and chloroplasts are both about the same size of bacteria. They both have their own DNA inside of them, and the DNA inside of a mitochondria or a chloroplast looks like bacterial DNA. It copies like bacterial DNA. Mitochondria and chloroplasts can copy themselves by binary fission, just like a bacteria would. I mean, they, by all intents and purposes, are little bacteria that have lost the ability to live on their own, that now just have this symbiotic relationship with our cells. Okay, you are able to tell me now, what is the structure of a nucleus of a eukaryote? How does that compare to the nucleoid of a prokaryote? How do rough and smooth ER compare structurally and functionally? What organelles are not associated with the Golgi complex? And we at no point discuss lysosomes. Did we lose lysosomes? Okay, well, let's talk about lysosomes because apparently we've lost that slide. So lysosomes are these little things that develop out of the maturing phase of the Golgi. And they develop enzymes in them that do very dangerous reactions. So lysosomes are where materials are broken down. If you have an old mitochondria, you send it to the lysosome to let it get destroyed. If you have, if you swallow a bacteria, if that cell engulfs a bacteria, it fuses it with a lysosome to break down that bacteria. So lysosomes are what are breaking down. They're kind of the recycling factory of your cell. Okay, so now we're back to the end of the lecture. If you have any question about eukaryotic cells, please let me know. We have finished all the material for your first exam. So I look forward to having you tell me everything that you've learned, everything that you've spent this time studying these past few weeks. And I will see you soon. Bye.